Now I'm going to demonstrate how we sample the epilithon, or what I referred to in the introduction as biofilm, the occurrence of a layer of living tissue on the surface of environmental surfaces in all these streams, being the rocks and the leaves and the gravel. Okay, this is the device that we use for sampling. It's called the lobe sampler, and the lobe sampler has at the end this little brush, and the idea is that you adhere the brush on an environmental surface. You pull it back so the brush is flush with the end point here, put it on an environmental surface, and then twist it to scrape what's on the surface off. And then this is a vacuum. You vacuum that water in and fill it up, and that water sample will then contain whatever it is that you scraped off the surface of the rock. The details of the structure are interesting. So, for example, it's got this collar of foam right here, and that's there so that it makes a good seal on the irregular surface that you're going to be adhering it to. And then the plunger has a rubber seal right here so that it makes a watertight seal against the length of the tube. And then it's got this little hole right here. And so what I'll do is I'll put my finger over the hole like this, and then I'll press it down on an environmental surface, crank the brush 5 times clockwise, 5 times anti-clockwise, take my finger off the hole, and pull up the plunger so that it fills with water. And so the water runs from the hole just into this surface, which means it's going to suck up everything that was scraped off of the environmental surface, and everything that was on the environmental surface will now be in this tube. And then I cut my hand over it and I save that sample. And it's that we use to get a sample of what it was that was actually on the surface of the rock that I'll be sampling. And this also represents the standard sampling area, so that the samples are fairly uniform from one to the next. Okay, now I'll demonstrate. Okay, so I put the brush so that it's flush with the end point, and I'm going to put my finger over this hole, and I'll press this against the surface of the rock and press down and turn clockwise 5 times, counterclockwise 5 times. Take my finger off the hole, pull up the plunger, put my finger back on the hole, and then lift it up. And you'll see that the water sample now is cloudy, and the cloudiness is all the sediment and other things that were living on the surface of the rock. And we'll save this sample and analyze it. We'll look, for example, at the amount of chlorophyll that's in the sample and in the other things that are present in the sample to characterize what's on the environmental surfaces in a natural stream. And we'll do this in streams that either have an intact canopy or a cleared canopy, and it's one way of informing us about what the effects of increased light level are on the stream ecosystem.